Hello and welcome to some biology class. Friends, today we shall discuss about toe printing assay. Toe printing assay, also known as primer extension inhibition assay, is a method in molecular biology used to study the interactions between mRNA and ribosome or RNA binding protein. This technique is used to study the mechanistic details of translation initiation of specific mRNAs in vitro. The overall goal of toy printing is to assess the ability of an mRNA to form stable complex with ribosomes under a variety of conditions in order to determine which sequences, structural elements or accessory factors are required for efficient translation initiation. For example, ribosome recruitment might be hindered in the absence of a 5' cap, but it is stimulated by the presence of an internal ribosomal entry site. This technique was developed by Gold and co-workers. Translation is the process by which polypeptide chains are synthesized from amino acids by utilizing the information stored in the codons of mRNA. It has 4 steps. First step is charging tRNA. Second step is initiation. Third step is elongation and the last step is termination. The first step in toy printing assay is in vitro transcription of specific RNA. This is the mRNA which has been transcribed in vitro, and we want to study the interactions between this mRNA and the ribosome. In second step, the mRNA is incubated with the cellular extracts containing translation components or purified components so that a stable complex will be formed between the mRNA and the ribosome. In this example, we have included cycloheximide in the cellular extract. Cycloheximide is an inhibitor of translational elongation in eukaryotes. Due to the presence of cycloheximide, elongation of translation will be inhibited, but initiation complex will be formed between the ribosome and the mRNA. In next step, a short primer, which can be radio-labeled or fluorescently labeled, is annealed to the 3' region of the mRNA. After that, the primer is extended by using the enzyme reverse transcriptase. It is because the ribosome is arrested, that means it cannot move on the mRNA. Because of this, reverse transcription results in the formation of shortened cDNA toy prints. And point is, when the reverse transcriptase encounter the stalled ribosome, then it is separated from the first base of the S side codon by 13 to 14 nucleotides. In another experiment, we have incubated the mRNA with the radio-labeled primer and the enzyme reverse transcriptase. In this case, reverse transcriptase will extend the primer fully up to the 5' end of mRNA. So, full-length cDNA will be obtained. But in this case, because the ribosome is acting as an obstacle, so the reverse transcriptase will not be able to fully transcribe the mRNA. So we will get shortened cDNA toy prints. Here we are getting two sets of cDNA fragments. One set include shortened cDNA toeprints that are formed due to the incomplete reverse transcription. When the enzyme reverse transcriptase encounter the stalled ribosome, the reverse transcription is stopped. So we are getting shortened cDNA toeprints. In this example, we will be able to know exactly where the ribosome is stalled along the mRNA. The second set include full-length cDNA fragments which are formed due to complete reverse transcription by the enzyme reverse transcriptase up to the 5' end of mRNA. The next step in this technique is to perform sequencing reactions, after which preparation of sequencing gels and electrophoresis are carried out. By comparing the bands formed by full-length cDNA and shortened cDNA toeprints with the sequencing ladder in the gel, we can determine the exact location where the ribosome is stalled on the mRNA. This protocol has an advantage, which is the use of a fluorescently labeled primer and a fluorescence gel-based imager. This eliminates the extra steps including radioactively labeling primer, drying the gel and exposing the gel to an intensifying screen. Because of this, fluorescence bands are recorded as the gel runs in real time that allow for greater resolution. So friends, this is all about Toeprinting assay. For more updates, please subscribe some biology class and also please share your comments. Thank you very much.